This is the second recording for the Biology EOC review. Strand 2 of biology is the mechanisms of genetics, and so the first question on your review sheet says label the components of DNA that are listed here and show the hydrogen bonds, and so I gave you this diagram to label. So here's deoxyribose, this pentagon-shaped structure with an O in the top of it is deoxyribose, which is the sugar in DNA. The phosphates are the PO4s that link together the sugars in the sugar phosphate backbone of the molecule. Adenine is one of the 4 nitrogen bases. Thymine is its partner here. Guanine is another one of the bases, and its partner here is cytosine. And these are hydrogen bonds that hold the nitrogen bases together to form the rungs of the ladder. So the sides of the ladder are made of the sugar phosphates, the rungs are made of the nitrogen bases that are paired together. The purines are adenine and guanine. Notice the purines have 2 ring structures, and the pyrimidines are cytosine and thymine, and so they have a single ring structure. Remember, adenine always bonds with thymine, cytosine always bonds with guanine. We always have a purine and a pyrimidine together. The way to remember this, ags are pure. Remember that? Ags are pure. It will help you remember that adenine and guanine are purines, and so the other 2 are pyrimidines. Now, there's a video that I will show that I will post separately for you on Edmodo, so that you can watch that. That goes over the DNA structure again. Next, we have a chart about mutations to fill in, and so here are the answers to the chart. Okay, there are 6 kinds of mutations listed here. Substitution, when one base is substituted for another, and what it causes, it might change one amino acid, it might not, depending on how the codon changes. Insertion is when you have a base inserted between 2 others. This will change everything past that point. It's a frame shift mutation. Deletion, similar to insertion, just the reverse. When base is deleted, it will also change everything from that point on. These 2 right here are major mutations because they will change the whole protein past that point. This one might change one, might not change any at all. And inversion and duplication and translocation are chromosomal mutations. This is when part of a chromosome breaks off and is reinserted backwards. Okay, and so it can cause a birth defect or a genetic disease. Duplication is when you have a part of a chromosome that's duplicated twice, and again, it's probably going to cause some kind of a birth defect or genetic disease. And then translocation is when you have 2 chromosomes that part of them break off and reattach to the wrong one. Can also cause some kind of birth defect or genetic disease. There's also a DNA replication video and a mutations video that I will post separately from this presentation. Next, we have practice in Punnett squares, and you're supposed to identify the parent gametes and the genotypes of the parents and offspring. So here's a basic Punnett square, okay, and the gametes are going to be the letters that are on the outside of the Punnett square. Each one represents a gamete. This would be an egg cell. This would be the other possible egg cell this mother could produce, a sperm cell, and the other possible sperm cell the father could produce. The trait that we're talking about here is pointed ears, which is not really a human trait. I just did it to be funny. And big A would be homozygous dominant. Big A, little a would be heterozygous. And so both of these parents are heterozygous for the trait pointed ears, meaning they would have pointed ears, but they also carry a recessive allele for that trait. If you have round ears, then you have little a. That's a homozygous recessive condition. And so the parent genotypes are both heterozygous, and the offspring genotypes, 1 would be homozygous dominant, 2 heterozygous, 1 homozygous recessive. So these parents would have 3/4 of their children probably would have pointed ears, and 1/4 of their children probably would have round ears. Next, we have explained the differences between transcription and translation. And there's a table here to fill in also. We have the 2 processes, transcription and translation. Transcription occurs in the nucleus, and it involves DNA, RNA polymerase, and nucleotides, and it produces messenger RNA. Translation occurs in the cytoplasms, on the ribosomes, and The molecules needed are messenger RNA, transfer RNA, and amino acids, and the product is a protein or polypeptide. And there is also a video on DNA to protein. The similar is the same one I believe that we saw in class that shows the actual molecules and how they form. Next, we will move on to strand 3, which is biological evolution and classification. And the first thing you're supposed to do here is list the types of evidence of common ancestry among the groups. So we have, first of all, homologous structures of living organisms. These are structures that are similar, or they have the same origins, but maybe have different structures and different functions. So that would be something like the four limbs of a cat or a dog or horse and your arms. They are similar in structure, they have the same kind of origin, the function and the actual shape of them are very different. Next, vestigial organs. Vestigial organs are things that are no longer useful in the organism, like your appendix is an example for one. Limb bones in snakes, when they don't have limbs, they don't have legs, but some snakes do have the bones for them. There are a number of other vestigial structures that you'll see in other kinds of organisms. Next, we have similarities in embryological development. When we look at the embryos of various animals at different stages, you remember they look very, very similar to each other in some of those embryonic states. Once they reach the embryo part, it takes a while before you can tell the difference between them. The fossil record is also another evidence for evolution. This is when you see the transition from one particular form to another in the fossil record. And geographic distribution of living species, so we can see that there are the same sorts of animals found in the same sorts of places. Some animal fossils or plant fossils are found in corresponding places in the same age of rock. And then finally, DNA analysis, which lets us do something like electrophoresis that shows the similarities and differences like we saw on the electrophoresis activity about the bare branch in the tree of life. There's also a video about evidence for evolution that shows some good pictures, and I will post it separate from this presentation. Next question says list the elements of natural selection. So these are the parts of Darwin's law, basically, Darwin's rules. There's a struggle for survival. Remember that organisms have to compete for limited resources. There is variation within each species. They're not all identical, and some variations will give some advantages. Some variations are better adapted to the environment, which means they have greater fitness. And those with greater fitness survive to reproduce and pass on their offspring, so that we have, over time, descent with modification from one form to the next. In the next step, we have defined the terms here about biological evolution. So we have adaptation, which are inherited characteristics that increase an organism's chances of survival. Adaptive radiation, which is when a single species evolves in different forms that live in different ways. It often happens when a group of organisms move into a new area that has not been exploited by their kind before. Convergent evolution, where we have unrelated organisms that live in similar habitats that come to resemble each other like a dolphin and a shark do. Next, we'll move on to taxonomy. So we're listing the taxonomic categories from largest to smallest. That is the domain, the kingdom, phylum, class, order, family, genus, and species. Remember, you can use the sentence, Dear King Philip came over for great spaghetti, or something similar to help you remember the order in which they occur. Next, we have characteristics of the kingdoms. So we have archaebacteria, which are prokaryotic, live in extreme environments. They do not have peptidoglycan. The eubacteria, which are also prokaryotic, they do have peptidoglycan in their cell walls. And protista, which are eukaryotic, that are not fungi, plants, or animals. Remember, this is the kingdom where you put everything that doesn't fit someplace else. And then fungi, which are heterotrophs with cell walls made of chitin. Plants, multicellular eukaryotic photoautotrophs with chloroplasts and cell walls made of cellulose. And then finally, animals, which are multicellular eukaryotic heterotrophs with no cell walls. Which kingdoms belong to which domains? We have domain Archaea, which entirely consists of kingdom Archaebacteria. domain bacteria, which is entirely made of kingdom Eubacteria, and domain Eukarya, which includes 4 kingdoms: the Protista, fungi, plants, and animals. There is also a taxonomy video, which I will post separate from this video. And this is the end of the second EOC review video.